Messed up genetic experiments. Genetic experimentation has been controversial since its discovery. Is it a force for good or evil? Will it reveal the secrets of existence, or should we live it well alone? Here's our countdown of the most messed up, bizarre, and controversial genetic experiments in history. Number 10. Human cow embryos. In 2008, British scientists managed to successfully grow embryos and stem cells using human cells and the egg cells of cows. The scientists hollowed out egg cells taken from cattle before inserting human DNA to create a growing embryo. They were then able to take the embryo apart again and collect embryonic stem cells that had grown inside.
Part of an elite of British researchers who've become world-renowned for their work into stem cells. Cells which have the potential of developing into any part of the body.
Stem cells are sometimes called the body's master cell, capable of changing into every other cell in the human body. The cells have extraordinary regenerative abilities and as such are extremely important to medical researchers attempting to find new treatments for dangerous illnesses. The ability to grow these cells inside the egg of a cow rather than a human was a massive stepping stone, as it allowed scientists to carry out new stem cell research without wasting valuable human eggs. Stem cell research has its critics, however, with some arguing that even the smallest fertilized eggs must be treated with respect. Regardless of what you believe, stem cell research is at the most accepted end of the scale when you compare it to some of the other experiments on this list. If you thought this one was shocking, you're in for a bumpy ride. Number 9. Human Z's. It may turn your stomach, but scientists have attempted to create a creature that is half human and half chimpanzee. Soviet scientists first tried it in the 1920s, attempting to artificially inseminate female chimpanzees with human sperm. The test failed to achieve a pregnancy, but inspired others to try decades later.
In countries that were underpinned by atheism, which the new USSR was, how better to reject religion than to be able to prove that Darwin's theories were correct.
The next attempt was by Chinese scientists in 1967, who claimed to have successfully impregnated a female chimpanzee with human sperm. The experiment was reportedly cut short, however, for political reasons, and the chimp died of neglect before giving birth. No further human and primate hybrid experimentation had been reported until 2019, when scientists in California produced the first human monkey chimera, an organism that contains the DNA of both human and a monkey. Development was stopped before the specimen developed a nervous system or organs to avoid ethical concerns, and was carried out in China to avoid legal issues. Who knows what would have happened if these experiments were continued? Do we even want to find out? Number 8. Glow-in-the-dark animals. In the last 20 years, scientists, and even one artist, have managed to use DNA from existing animals to make other animals capable of glowing in the dark. In 2012, scientists in Uruguay injected several sheep with green jellyfish protein soon after their birth. As the sheep grew up, they acted like regular sheep, except they gave off a greenish glow when exposed to certain UV lights.
In the dark, under fluorescent light, is when they show their true colors. These are fluorescent sheep.
In 2008, scientists in Taiwan created pigs that glowed from the inside out by injecting pig embryos with DNA from fluorescent jellyfish. The 3 piglets that were born had a slight greenish tint during the day, but glowed bright green in the dark when exposed to blue light. They also not only had fluorescent skin and eyes, but also glowing organs. You don't want to know how scientists figured that last part out. Finally, artist Edward Cax showed in 2000 that anyone can have a go at producing glow-in-the-dark animals, although they probably shouldn't. He successfully created a rabbit that glowed when exposed to blue light. The actual experiment was carried out by French scientists who injected jellyfish proteins into a fertilized rabbit egg.
And she looks like any other plain old white rabbit. Well, that's until you put her under a blue light, at which point dear little Alba begins to glow green.
Despite Cax's desire to take the rabbit home as a pet, disagreements broke out over the ethical implications of this so-called art project. Sadly, Alba the bunny died 2 years after her birth, bringing the debate to a sudden end. Number 7. Belgian Super Cow. We've all heard about how breeds of dogs were deliberately created by humans. Selective breeding allowed us to turn vicious wolves into the trusting, floppy-eared friends we know today. However, did you ever wonder if humans tried to create the ultimate fighting animal instead? Well, they kinda did, but with cows. In the 1800s, Belgian farmers began breeding local Belgian cows with a shorthorn breed from the UK. They mixed in a few others too, choosing the biggest cows from each generation to breed each time. Eventually, they ended up with a new breed called the Belgian Blue, which is much more muscular than your average cattle. The breed's purpose was never intended to be for fighting or utilizing their strength, however, as they are instead valued for producing meat that has a reduced fat content and reduced tenderness. Number 6. Editing human DNA. In November 2018, the scientific world was shocked when Chinese biophysicist Zhang He announced that he had successfully edited the DNA of human embryos before they were born. He claimed to have successfully made 2 baby girls immune to HIV by editing their DNA before birth. The 2 girls, Lulu and Nana, are reportedly living happy and healthy lives, but the tests were widely condemned by the scientific community.
A line has been crossed that should not have been crossed.
It's very disturbing. It's inappropriate.
Oh, this is huge.
Zhang He was subsequently sacked by his university and given a 3-year prison sentence for carrying out this risky and premature research. He later admitted to have acted too quickly, but refused to say that he regretted it. While gene editing in adults is being looked into around the world, his experiment was the first time embryos had been altered. It meant that the changes made would be passed down to future generations if successful. Other scientists pointed out soon after that Zhang Kui's work would be used to increase intelligence and memory in unborn children. Could one man's research have revealed the key to creating genetically modified superhumans? Is that even something that we would even want to happen? Regardless of what you believe, no further experiments are officially being carried out today. Number 5. Reviving extinct animals. This one may strike fear into many Jurassic Park fans out there, but don't worry, there aren't any plans to bring back dinosaurs. Yet. There is, however, serious research going into the revival of other more recently extinct animals. Some scientists now believe that certain species could be brought back from extinction in just a few years if one biotech company is successful. Colossal is a company that has plans to bring back species including the woolly mammoth, the dodo, and the Tasmanian tiger.
The DNA technology involves taking cells from the tiger's closest living relatives and splicing them with Tasmanian tiger DNA.
The company plans to use gene editing technology to insert extinct woolly mammoth DNA into the genome of an Asian elephant. Because mammoths and Asian elephants share over 99% of their DNA, the scientists plan to have the first woolly mammoth calves by 2028 and have promised to reintroduce them into the wild. The company is doing similar work with other species also. Last year, the bioscience company announced it had raised millions from investors hoping to support the research. So keep an eye out for new species roaming about pretty soon. Number 4. The Vacati mouse.
On the back of this animal, we're actually incubating and growing perfect cartilage in the shape of a human ear.
The 1996 Vacati mouse was a laboratory mouse that had what looked like a human ear grown on its back. Created by scientist Charles Vacati in 1996, the ear was actually cartilage grown from the cells from the knee of a cow in the shape of a human ear. Scientists inserted these cells into an ear-shaped structure and then implanted it under the mouse's skin. You might be one of those people who find mice creepy already. Now, have one that has no hair and a human ear growing on its back. The idea was to see if it was possible to grow cartilage artificially that could be reused elsewhere. The experiment was a success, and the mouse began to grow extra blood vessels that nourished the new cow cartilage ear. Over a few months, the structure that the scientists placed in the mouse dissolved, leaving a healthy ear-shaped piece of cartilage under its skin that could be removed and used elsewhere.
So when this finally gets implanted in a human, you don't expect rejection, as is so common with new body parts.
Exactly, because we're going to start with the patient's own cells. It'll make his own tissue, and therefore the body will accept it.
It's possible that one day this method could be used to grow working organs, with some scientists suggesting that artificial organs may be used to save lives in the next few decades. Others said that sacrificing a few mice was worth potentially saving lives, especially as most people are happy to sacrifice a cow just so that they can have their lunch. Number 3. Lab-grown meat. One of the most recent developments on our list, lab-grown meat is something that we may find on our supermarket shelves sooner than you think. In June 2023, the U.S. Department of Agriculture gave one company the green light to begin producing and selling chicken that doesn't come from chickens. So how is it made and what is it really like? Lab-grown meat is meat that is developed from animal cells and grown in massive bioreactors. Building on the genetic research carried out in some of the other experiments, scientists are able to create the perfect conditions for these cells to grow into full chunks of tissue. Although many people find the idea of growing meat in a vat to be slightly odd, the potential benefits are huge. Not only does it remove the need to slaughter animals, but it would free up a huge amount of land and water usage needed for traditional animal agriculture. The important question for many, however, is how does it taste? It seems that it's not bad, with reporters and other tasters reportedly giving decent reviews so far.
It looks totally normal. Here goes. Like, I want to give some incredibly scientific response to this, being a scientist, but like, it's a crunchy, normal piece of chicken.
The environmental devastation caused by humans' consumption of meat has been known for decades. Could this be an important step in the battle against climate change? Number 2. HeLa cells. In 1951, Henrietta Lacks was being treated for an aggressive case of cervical cancer at the Johns Hopkins Hospital in Baltimore. While treating her, doctors harvested some of her cancerous cells and gave them to researchers without her consent or knowledge. Researchers in the lab discovered that Henrietta's cancer cells had an extraordinary ability to survive and reproduce.
All human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor. Dark purple, shiny, jelly-like.
They were effectively immortal. The researchers shared them with other scientists to be used in a wide array of scientific experiments. Henrietta's cells were so useful because they were extremely robust and reproduced much more quickly than regular cells, doubling in number after about 13 hours. It is estimated that 50 tons of these so-called HeLa cells have been produced to this day to be used in research around the world. About 75,000 scientific articles were made possible by using the cells. HeLa cells were also used to create the first polio and cervical cancer vaccines, as well as numerous other scientific breakthroughs. Despite their use, Henrietta was never asked or informed about any of this experimentation before her death. Her DNA continues to be used around the world to this day, with some arguing that it should be stopped. Number 1. Cloning.
On an ordinary farm in Scotland, scientists say a clone was created from a single cell taken from the udder of a sheep. The embryo was then implanted in a surrogate, making an exact genetic copy of its so-called mother.
You may know Dolly the sheep as the first ever cloned animal. It may surprise you, however, to know that this isn't true at all. The first clone was actually a sea urchin that was created in 1885 by German scientist Hans Driesch. He discovered that separating sea urchin embryos caused two fully genetically identical urchins to form rather than two halves of one animal. His experiment was the first of many to artificially form an animal that was genetically identical to another. There were many more successful cloning experiments that led to Dolly's birth in 1996, and she certainly wasn't the first. Sorry, Dolly. The first ever cloned mammal was a sheep born in 1984 that was grown from a part of an embryo and placed inside a surrogate mother. Two other sheep named Megan and Morag were the first mammals to have been successfully cloned from cells that weren't stem cells. The reason Dolly was so famous is that she was the first clone to be made using an adult cell. The implications were huge, as it was discovered that day that all a team of scientists would need is a tiny amount of your DNA to clone you in a laboratory. Creepy. Dolly and other cloned sheep also lived happily and healthy lives, adding fuel to the discussion.
Cloned sheep using the same method as that which created Dolly showed no obvious detrimental long-term health effects.
Dolly's birth and healthy life began a conversation that continues to this day. No one has ever attempted to clone a human being due to the legal and ethical concerns of doing so, but some believe that humans will attempt it one day as the study of genetics continues to advance. So what do you think? Is genetic research a tool for good, or is it something that humans should never have messed with? Are there any cases we missed out? Let us know in the comments below. We hope you enjoyed this episode of History 101.